Recording in progress. Recording stopped.
When you go on Canvas, do you see this Zoom meeting? Can you check on your student Canvas? Do you see the Zoom meeting for class today? I just want to see if I need to send it separately to check time.
Yeah, there is a recurring meeting.
Okay, good. So hopefully he finds that. Right.
It's just
Wait a couple minutes. See if he can email me. What is this showing? What's it gonna do? Hello? So sometimes this kicks me out and doesn't let me share my screen for some reason. It's very annoying. But I just posted the notes for the for today and maybe next week. I'm trying to make this share my screen here. Let's see, let's see what check time and He did not email. Does anyone know if I did get a thing that he might be absent from Starfish. Oh. I was missing. Oh. Oh, sorry. You're not supposed to be reading that. Ah! You're not supposed to see that.
Sorry.
That's okay. It's my fault. I had it up there. I guess he might be sick. So, okay. Okay. So I guess we can just get started. If he emails us or something, we'll see.
He said yes, I joined via link that
He
was sent earlier.
he joined.
Via some link that was sent earlier. So should I just let him know that he should join
On Canvas, yes. Mm-hmm. Yeah.
Two
okay.
participants.
What's that?
The two participants.
That's me. Both of them are me. Me up there and me over here.
He's joined.
Okay. Did Dr. Johnson usually start the video up there? So, do you know?
He used to, but then he kind of collapsed. Like he used to collapse the video.
Oh, okay. Hi, Titan.
Hello,
Can you hear us?
I can hear you. Yes.
You can't see us. Did Dr. Johnson usually put the video on so you could see us? Would you like that on?
No, that's all right.
Oh, okay. All right. Well, we're all here. What I wanted to do is start reading some of these chapters in the book. It's a book about it's called Signal Transduction. And I thought this would be nice to complement what Dr. Johnson had already told you, because what this book does is it goes a little bit deeper into all the topics that Dr. Johnson covered, right? So hopefully you got a chance to read through some of that chapter before today. I was just going to go through and summarize the chapter and some of the points that I thought were important, and interject whenever you like if there's something that you read that you wanted to talk about in the chapter. So the first thing they talked about is, we started with G protein coupled receptors, or Dr. Johnson did the first or second week, talked about G protein coupled receptors. So we all know at this point, right, that G protein coupled receptors are 7 transmembrane spanning, they have 7 transmembrane spanning domains, and that's very characteristic of the G protein coupled receptors. And they have a extracellular ligand binding site, right? And then an intracellular C-terminus, correct? So we're all on board with that. And then we know that they bind to these G proteins, also known as GTP binding proteins. And which are the G proteins that Dr. Johnson told you about? How many did he mention to you?
He said like three we know, like for sure, like that will be the focus of the class. But then there's one type that he said like we won't discuss much in detail. That's I guess GQ or something.
Yeah.
GQ. He said like,
GI, GQ, G alpha, G.
So he talked about
whatever,
GS,
G12.
GI, and GO, and GQ, right? Yeah. GI, GO are often put together. So what I like about the book is, I know he said he'd focus on those, but I think it's important to know that those aren't the only 3, right? So the book goes a little bit more into detail about more subtypes of G protein, which I think is important to understand that there's more, especially at your level, because you might come up, in your research, you might come up against a G protein coupled receptor that doesn't fit into one of those boxes,
Right.
the GI, GS, or GQ. So you should know what's going on, and maybe you'll discover a new G protein. Who knows? All right. Okay. So, and I also like this book because it gives a little bit of a history of how these things were discovered. Right. So the first thing it talks about in Figure 4.1, which I can pull up here. Hold on a second. I have to go back to my box if I can. Okay. So let's I'll pull up the chapter here. So in this first they established the importance of these nucleotides as metabolic regulators. So not just GTP, but there's CTP, obviously ATP has a lot of functions and stuff. But then it goes into how these GTP binding proteins were discovered. So at first, when they were studying these G protein coupled receptors, shown here, sorry the copies aren't the best, but it shows that they had a receptor, and then all they had to do was add ATP to their preparation, and they got the reaction to occur, such that the amplifier or the effector, which is, as we know now, the enzyme, right? That's a adenylate cyclase enzyme. What they found is that the ATP would be converted to cyclic AMP. They didn't need any other thing other than the receptor and the effector enzyme, right, in that prep. So for a long time that worked, but then what happened is as they got better and better at manufacturing ATP, it became more and more pure, right? So what happened in the beginning, they think, is that the ATP was, since it was not purified, it was contaminated with the G protein, right? So there was some of the G protein there, so all they needed to do was add in the ATP along with the G protein, and they could get the reaction to work. But over time, as that became more pure, right, they weren't able to get that to work. So they reasoned there has to be something else in between those steps that allow the reaction to occur, right? So the impurity was the GTP, actually, because the G protein would be part of the receptor. The GTP was coming along with the ATP, so that the GTP could be used by the G protein to be activated and and allow the enzyme to convert ATP to cyclic AMP. So once they figured out the impurity was GTP, they realized that's when they realized there must be some kind of protein binding to this GTP that's activating these receptors, right? So one thing I thought was interesting too about the GTP binding proteins, we call them G proteins and GTPases, right? We make that distinction. So the G proteins are the ones that we normally think of, right? The three subunits, alpha, beta, and gamma. But the GTPases are the ones that are monomeric. But this is going to be a pain. This is really a misnomer because, sorry, my computer does this. They said it's a connectivity issue. I just gotta keep, I have another class where I have to keep going back and doing this. Okay. Right, but it's really a misnomer because all of them are GTPases, right? We understand that they all have the intrinsic ability to take GTP and convert it to GDP, right? But I think for us to keep them clear, we keep them separated so that we know that the G proteins are the ones that are the heterotrimeric proteins, whereas the GTPases are the monomeric GTP binding proteins like RAS and RAF, those family of oncogene. Well, RAS is a proto-oncogene. So, does everyone know kind of about RAS, how it works?
RAS,
Snap, like
Then-
using the MAP kinase.
Right.
there are 3 times phosphorylation to. Yeah, I think you explained it.
Yes, right. Yeah, I think I talked about it a little
Yeah.
bit. Right. So we have. Is this in? Okay. So we have for this we have RAS, RAF. Right. Then goes to the MEKs. Then goes to the MAP kinases. Right. So why we call RAS a proto-oncogene is because when RAS is mutated in cancer, right, RAS becomes constitutively active, and so since RAS acts like it is a GTPase, and it acts like the conventional G proteins that we know, the heterotrimeric ones. It's activated downstream of those receptors. Typically, RAS is activated downstream, though, of the kinase linked receptors, right? So we have the kinase link receptor activating the RAS, then the MEK, then the MAP kinase. But in cancer cells, a lot of times this RAS is mutated such that it no longer needs the activation of the receptor tyrosine kinases. Right. Okay. All right. So I just wanted to make that distinction. For our purposes, and actually for whenever you're reading about G proteins, you should think, when you hear G protein, you should think that alpha, beta, gamma. When you hear GTPases, you think of the monomeric RAS. Right? So then it gives you a picture in the book of the structure, and it shows you that the alpha subunit and the beta gamma subunit. And let's pull up that if I can. Let's look at the structure for a bit. So I have to keep going back and forth. Okay, let's find it. I'm looking for it. Yes.
Whenever we hear RAS, RAF, and MAP kinase, it always comes up with tyrosine kinase receptors.
Right.
But when we are talking about G protein coupled receptor, we always think about alpha, beta, gamma.
That's correct.
Protameric. So, ma'am, what's the difference of the RAS over here and the RAS in the tyrosine kinase receptor? Are they same or? Are
That's
they
the
different?
same. No, this is the RAS we're talking about.
Yeah.
Right?
Yeah.
So, I'm sorry, am I missing?
No, I was just asking that, is this the same thing? Because whenever this RAF comes up, we normally think about tyrosine kinase
It's
receptor.
connected.
Right.
And here we are in G protein coupled receptors, in GTP.
Right,
Things we
right.
are talking about RAS and RAF.
Right.
So are these both the same things? That's what I was asking.
Yeah, so in the chapter, they are just talking about GTP binding proteins as in general, right? And then they talk more first about what the classic GTP binding proteins that we think of, the ones that are coupled with the 7 transmembrane spanning receptors, the GPCRs. But then RAS and RAF, they consider separately, although they're all GTP binding proteins, right? And the RAS and RAF are usually associated with the receptor tyrosine kinases. Yeah.
So Dr. Kamna, the RAS here appears to be playing a role of GTPase.
Mhm.
Like it is again a GTPase,
Yes.
but not associated with the receptor, but somewhere soluble in the.
Yeah. So it gets it gets recruited to the receptor tyrosine kinases, but it's not associated with them from the start, right? Not like that is that what you were asking? Yes, yeah. And actually, the way we think about it being associated with the receptor from the start is a little bit not incorrect, but we'll see. They show the cycle.
Misguiding maybe.
Misguiding
We have
maybe.
attached one thing with one thing. and
Right,
that's not
right. And
The
so
same.
we often think it's like one receptor, one GTP,
Yeah.
or one G protein, right? And that's it. And once that's done, you need another receptor and another G protein. When in reality, as they show in this chapter, we'll talk about that in a minute, the G protein actually dissociates from the receptor and then the G protein can go on and associate with other receptors, right? So that G protein can kind of be recycled. Okay. This is going to keep coming out. Sorry everyone. Sorry, Chetan. My screen keeps. Oh, it's up there yet, but it's not here. I hope the signal gets stronger. Okay. All right. So I just want to look at the structure here that they showed in the book. So it's hard. It's not in color, unfortunately, so it's hard for you to see. But you can see that the alpha subunit is there on the left, right? And then the beta gamma subunit is on the right. And the two are coupled via these two regions they call the SW1 and the SW2 loops. It shows you the structure of that beta gamma really looks like, they call it a helicopter, like blades of a helicopter if you're looking down. So this is as if you're looking, as if you're looking down into this like hole here. So if you were hovering above, you'd see that structure. And then talks about how the attachment to the membrane occurs through the N terminal of the alpha subunit and the C terminus of the gamma subunit. And it's hard to see because of the way this is situated, but they're talking basically about this terminal N and this C terminus here. Right? And that's how they attach to the membrane. Okay. Sorry, I keep doing that.
So, Dr. Kavanaugh, will
Yes.
it be right to say that the 7 transmembrane spanning part of the receptor is actually the beta gamma subunit?
The transmembrane spanning region? No. Of the receptor? No. So the receptor is its own thing, right? So we're going to have the receptor with the, I'll draw it on here. So we'll have the receptor, this is really thick here. That's the cell membrane, right? We have the receptor here. Right. And then the G protein is going to be very closely associated with this, and the C terminus here of the alpha and the N terminus of the beta gamma. So this would be G alpha here, and then beta gamma over here. Of course, they're all connected, right? And that's how they're anchored to the membrane. So they're not, those transmembrane domains of the receptor are separate or distinct from the G protein, right?
That's right. Like it's literally the name G protein coupled.
Yes, G protein coupled receptor because they're closely linked to each other. Right. Sorry. So it's the N. I have that wrong. Actually, it's the N terminus of the alpha loop and the C terminus and C here, right, of the gamma loop. Sorry about that. Okay, so. This is where I think it really challenges what we normally think about with the GTP cycle that we were talking about. The interaction with the receptor is actually transient. It's not one-to-one. It's not one receptor with one G protein, and then once that reaction is done, it's gone. And we call this kind of coupling collision coupling. So what happens is the G protein actually remains associated with the effector. So what's the effector? Does everyone know what the effector is?
The adenylate cyclase.
The enzyme adenylate cyclase or phospholipase C, right? Depending on, and then other enzymes for the other G proteins. And the way that we can keep these cycle going is that there's a lot of GTP in the cell, actually about 10 times more than GDP in the cell. So it allows the G protein to become inactive, the GTPase to hydrolyze the GTP to GDP, but then also there's a lot of GTP around to come in and exchange there. So the way we think about G proteins then, because they have this GTPase activity, is we think of them as like switches. And I like how the book explains this, because it's not all, in some cases we're going to see, like in the visual system, the G protein, it has to turn off and on very quickly, right? But in other cases, it doesn't necessarily turn off and on. So when you think of a switch, you think off, on very quickly. But sometimes it takes a little bit of time. And how the book describes it is, it talks about if you have a light on upstairs, right, and you need to turn it off, you have to go up the stairs to the switch to turn it off. And that's kind of how it is. There's a little bit of time for the reaction, so the switch doesn't just go off right away. It's like kind of going off as you're going up the stairs, and once you get up the stairs, it's completely off, right? So there's some time where things are switching from on to off. And the way this activation is regulated, it's obviously a kinetic process. So how we can determine the activation ratio is by looking at the rate of GDP dissociation, so the GDP coming off of the alpha subunit, right? So the ability of the GDP to come off the alpha subunit. Yes, and I like that figure you're looking at. Young, I'm going to pull that up here. Yeah, so here, right? So we start with the ligand that's shown in the upper left here. Here, here, that's the ligand, right? And then we have the inactive. I hate this keeps coming up. Why does this keep doing this? This little bar here. Okay. And I'm gonna, you know what? Hold on a second. I want to go to notability. I think that's better. Sorry. Give me 1 second. Sorry about that. Here it is. Okay. This is better. I can write better here. Okay, so we have here the ligand, right? And the inactive G protein coupled receptor. Okay. And so what happens in this is that the ligand actually activates the receptor, and then you have an inactive G protein, but it's already associated with the effector. So that's another thing that I think is different than what we think of. We think of that always the alpha subunit goes off and binds the effector, and the beta gamma subunit goes off by itself. That's not always the case. A lot of times the beta gamma subunit goes to do its own thing, but sometimes they actually stay together, like in this picture. And so the GDP comes off, and the GDP, they have the exchange for GTP, right? So now you have an activated G protein and an active receptor, right, coupled together. And then what happens is this reaction right here, this interaction here, is actually pretty quick. So the receptor and the G protein, once they come together and they're both active, they dissociate pretty quickly. So now that active receptor can go off and join with other G proteins, right? And so the G protein then is free to activate the enzyme, the effector enzyme. It has its GTPase activity here, where it hydrolyzes the GTP to GDP. And now there's this inactive G protein looking for the next active receptor, right? And sometimes it can even reassociate with this same receptor if it stays active, right? So this is a way to recycle the G proteins. It also shows you that not only does the receptor interact with the G protein and determine the rate of the signaling, but the G protein also has some role in determining that rate of signaling, right? As the rate of the GTP being exchanged for, or GDP being exchanged for GTP, and the rate of the GTP hydrolysis plays a big role in how this signal transduction pathway is going to go. So the way we look at that then, is These didn't, but we can determine then the activity ratio by looking at the rate of GTP coming on. So that would be the concentration. This is supposed to be alpha. Sorry about that. G alpha GTP. So what's the concentration of the active G protein over the concentration of the inactive G protein, and the inactive G protein has the GDP, right? And what we call, these are constants that we call the dissociation and the catalytic constant. So the ability of the GDP to dissociate and exchange with GTP is called the dissociation constant. And the ability of the G protein to hydrolyze the GTP to GDP is called the catalytic constant. So at any point in time, if we wanted to figure out the activation of that G protein, we would have to determine this ratio.
Is it different for each G protein?
Yes, so a lot of it also depends on the effector enzyme that is the G protein interacts with. So sometimes it's a lot, the GTPase activity for whatever reason is faster when the G protein interacts with phospholipase C, for example. That reaction is faster than with adenylate cyclase. So I don't know why. I'm sure someone who studies G proteins like their whole life knows why, but there's something with the enzyme and how the effector enzyme and how it interacts with the G proteins that can change that rate of that catalytic rate. Yeah, so it's not the same for all of them. Some of them are faster and some of them are a little bit slower. So that's, let's see what else was in the chapter. I want to make sure. Yeah, so here's this equation, right? There's the constant of dissociation of the catalytic constant. And that's how we can determine the activation of these G proteins. So I also like in this book, so you talked about agonists versus partial agonists, like full agonists versus partial agonists. So a lot of times we think of that as the binding to the receptor. Maybe the binding to the receptor is not as strong or something, but actually that can be true. But in some cases, if you read this side note here, right? Some cases it's actually you can ascribe this partial agonist ability as lower rates of the GDP dissociation, right? So instead of having the GDP dissociate and GTP coming on, if when these partial agonists bind, instead of making that reaction maximal, what happens is it lowers the rate of the dissociation of GDP, which is really cool. I think that's really cool to think about, that something can be a partial agonist, bind to the same receptor, but whatever is causing those interactions between the receptor and the G protein is just holding that G protein in that inactive state a little bit longer than not. Let's see. Right, so the partial agonists, what are happening is they can't distinguish as well between the active and inactive conformations of the receptor. So, did it show it here, or is it, oh, it's earlier in the book. Oh, no, here it shows, no, that's the GTP gamma S. I don't know if I have that page 66. Let me see if I have that in my notes. I think I have the whole book scanned in. Oh no, it's locked up again. So then actually when we think about antagonists, right, the antagonists are different. They just block the receptor. They don't allow any kind of reaction to occur. So G proteins don't regulate that as well, as don't play a role in regulating that effect of the antagonists, right? Because the antagonists are just changing the receptor such that there's there's actually that interaction between the drug doesn't even occur. But the partial agonists are causing that that delay because the partial agonists will bind. They'll bind here to this inactive receptor, and they don't distinguish between the inactive and active receptor. I wish I could pull this up, but it froze on me again. Oh no, it didn't. Why is this not working? Why won't it let me go back? Ugh. Okay, sorry.
You have to zoom out first and then.
Oh, maybe. No? Oh. Okay. Thank you. So, trying to see if I have that page in what page did it say? 66? Oh no, this only goes to page 44. Wait, how's that possible? Sorry, I'm looking for page. Oh wait. Oh, this is Goodman and Gilman. That's a different book. Yeah, sorry, I don't have that page here, but I'll find it and I'll post it for you. Okay. Oh, no, we're still on it. Here we go. Okay, and then the other thing. It's just delayed. So far, right, there's several steps of regulation, right? The G protein itself, how fast it dissociates the GDP and how fast it hydrolyzes the GTP. At the level of the receptor, if it's a partial agonist versus an full-on agonist, so the receptor is communicating to the G protein, but the G protein is also regulating and communicating to the receptor. But then there's also regulators of G protein signaling and GTPase activating proteins that can also modulate the signaling of the G protein coupled receptors. And for the regulators of G protein coupled signaling are the RGS proteins. There's smaller versions of this, smaller proteins that are less than 220 residues, and then larger ones that have a lot of different domains. And then we have the GTPase activating proteins. And the book goes on to break these down further. So, okay. So, this is in the next note page. And okay, anyway. So looking back on that figure, we talk about the contact between the alpha subunit and the effector, because that's the alpha subunit is what is binding to the effector and causing that effector to become activated. So when that contact occurs, that then is the signal for the GTPase reaction to occur. And as soon as they bind together, that accelerates the activity of that GTPase, right? So, well, that means that it's either an increase in the catalytic constant or the dissociation constant. Which are we talking about in this case? The GTPase reaction.
GTP is dissociation of GTP.
The other
One.
The first one.
Catalysis, right? So it's going to be the catalytic constant is going to increase when the G proteins, the alpha subunit contacts the effector. Okay, so I just said that there's regulators of the G protein. There's about 20 members of the RGS G protein signaling family. I said they're small and large, and then those are also subject to regulation. And so those would and then there's also GAPs or GTPase activating proteins. So if those are going to increase the K catalytic, and the regulators of G protein coupled signaling can affect both constants, right? The dissociation constants or the catalytic constants. So there's a lot of regulation of these G proteins, which makes sense, right? We don't want them to go unregulated in the cell. We want there to be regulation of the G proteins. And then we were kind of talking about this a little bit already, but the receptor affinity for the ligand is also modulated by G proteins. So you have regulation, again, at the level of the receptor and at the level of the G protein. So let's look at those figures, figure 4.4 and 4.5. We'll get to that in a minute. Where's 4.4? Okay. Yeah, so how this was determined is they found stable analogs, structural stable analogs of GTP, and they're shown here. The most common one is GTP gamma S, and this is used a lot in laboratories, right, to activate G proteins and to look at the result and how you can displace that GTP gamma S with when you add ligand or when you add GTP. So these analogs will not become easily hydrolyzed to GDP, right? So they stay in their active state. They stay bound to the alpha subunit, and they keep the G protein activated. I'm just trying to think about what I was trying to say here. So when an agonist binds to the receptor, it's going to signal to the G protein, and that, since I told you that's a very transient reaction, as soon as the activated receptor with the agonist binds to the G protein, and it dissociates, that is the signal for that GTPase activity to occur. See if it stays. Okay, it stays. So that's the way the receptors speak, quote unquote, to. I'm really sorry. Speak to the G proteins, and then the G proteins sorry. Sorry. Come on. It looks fine up there, but it's not letting me share on this screen. Sorry, Chetan, this keeps freezing on my end. I'm trying to make it come up. So anyway, so we can think about this. So let's think about how this works. So the G protein, the receptor's activated, like we showed in that figure, by the ligand. It binds to the G protein. Okay. All right. And this is figure back here, right? We looked at what's happening here. So the receptor is obviously talking to the G protein here once it becomes activated, right? So there's regulation by the agonist binding to the receptor, and then because of this discovery of these analogs that do not become hydrolyzed, we showed that the G protein can regulate the receptor by altering this rate of GTP hydrolysis. Does Does that make sense? Right, so if it, go ahead.
So Dr. Pravina, in that case, receptor activation and GT-base activation occurs more or less at the same time.
Receptor activation and what?
And the GT-base activation, like they should
It's
be
fairly, yeah, they're about the same time, yes, right. It's fairly quickly, yeah. So, but, trying to make the point of what the book was saying here. So it was saying here, right, so one of the earliest indications of a role of GTP in the activation of the receptor mediated process was a reduction in the affinity for activating ligands by stable GTPs. Okay. So since the GTP, I'm trying to understand how they are talking about this. The GTP is not being hydrolyzed, right? It's stable. So the activated receptor is going off, and it just keeps looking. Let's see what it says here. Okay. So because these analogs are not readily hydrolyzed, they ensure that the alpha subunit remains persistently activated. Okay, so that means, then it says, this coupled with the observation that the application of agonist, but not antagonist, ligands accelerates the rate of GTP hydrolysis. So that we understand, right? The agonist binds, it increases the rate of the GTP hydrolysis. Gave the clue that the communication between the receptors and the GTP binding proteins is a two-way affair. The receptors speak to the G proteins, that makes sense, and the G proteins speak to the receptors. So following activation, the contact between the G protein and the agonist receptor complex is weakened. That's what we saw. So allowing the receptor to seek out another G protein molecule. And then the situation with the inverse agonist is the converse, which we'll look at that figure. So I guess I'm a little confused in how they're talking about this. So maybe you can clear it up for me. So we understand that the receptor is binding to the agonist, right? And then that associates with the activated G protein. That's transient, so the activated receptor goes off, as we saw up here, right? And this then that triggers this hydrolysis very quickly, okay, to GDP. But if we have a stable GTP here that does not, this essentially, this does not occur, right? So I'm guessing, how does that indicate that the G protein is speaking to the receptor? I'm confused about that now. I thought I understood it when I read it, but now I'm confused about it. So what do you think, Young?
So when I looked it up, the equation, active, inactive, on and off, the concentration wise, the equation, so the denominator will be goes up and goes down. I think something
Catalytic
related,
constant, right?
yeah,
Yes,
something
yes.
related with one. I think
So
that's
if
why
you
it's keep
don't
activating.
have the GTPase, if it's not being catalyzed, that means that, yeah, go ahead.
So in that case, when they say receptor is speaking to the G protein, that only means when ligand binds to the receptor, that thing is transduced to the G protein and the GDP is knocked off.
Right.
And then when the GDP is knocked off and GTP like associates with the protein, the G protein tells the RAS-like domain in the receptor, RAS-like domain, saying that like it activates basically the GTPase immediately. Kind of asks the receptors to kind of set free the ligand, activate the GTPase.
Right.
So it's two-way communication between
Yeah.
the receptor and the protein.
So say again. So where's the G protein communicating with the receptor?
So when we saw that structure, like initially in the very beginning, they had this RAS-like GTPase in the structure itself.
Here? Yes. RAS-like domain. Yep.
Right. So appears like that is GT base,
Yes.
and it is kind of like what I'm assuming is that particular domain is causing the activation of GT base, which is dissociating GTP in the which is attached to the beta gamma portion, the alpha
Right.
subunit of the
Mhm.
protein.
Yes.
And when GDP, so that GTP now becomes GDP,
Mhm.
it's exchanged with GDP.
Here.
That communicates the receptor to set off so it separates now. Once GDP is attached to the protein,
Mhm.
it dissociates with the receptor basically, and then tells the receptor basically go and search for the next G protein.
Right. Oh, okay. All right.
So
So that's where the communication comes in. So, but then if you keep it here in this stabilized,
So what I'm understanding is the G protein is coupled with the receptor only when it has GTP. Once the GTP is out and GDP is in,
it quickly
again
dissociates, right?
And then
Okay. So okay. Right. So this is going to go search out one of these inactive ones. Right. So by keeping this active, this kind of thing's not going to happen. They're not right. Okay, I see what you're saying. Yeah. Okay. I understood it when I read it. I thought, but now then I confused myself. Okay. So right. So the receptor is speaking to the G protein. Because once it's active, it seeks out that GDP bound protein. And then the G protein is speaking to the receptor, right? Because once it's bound to the GTP, it tells the receptor to just dissociate and look for the next one, right? Okay, good. Thank you. I totally got myself all confused there. All right. So, and this was aided by the finding of these stable GTP analogs. All right. So then it showed you how in this figure here what's happening with an inverse agonist. Right? So here is the this is showing the equilibrium between the inactive. So this is going to be the inactive receptor here, and the active receptor on the right here. The agonist ligands are going to bind exclusively to the active form of the receptor. Agonist ligands. So they look for the active form of the receptor and increase the amount of the receptor plus the ligand that can transmit a signal. So this increases here the amount of active receptor. Conversely, inverse agonists bind exclusively to the inactive form of the receptor and thus increase the total amount of the inactive receptor. It keeps the receptor inactive. So you have less of this active form able to transmit a signal. So that makes sense, right? And then we just showed that this active analog of GTP gamma S decreases the affinity for agonists and enhances the affinity for the inverse agonists because it's going to have those, it's not going to be able to bind again to these G proteins because it's looking for those inactive G proteins. So there's going to be more inactive receptor around and enhances the affinity for these inverse agonists. Oh, it's so annoying. And I lose my thought when it does this, and then I get. Okay. There we go. That's not what I want. Sorry. All right. So, basically, this whole first part, or this whole first handout for today was just showing you the different ways, the structure of the GTP binding proteins, right? How they bind via the C terminus on the gamma loop and the N terminus of the alpha loop. And then you're talking about the GTPA cycle and the ratio of activation. And then we moved on to this. Okay. All right. So now we're going to add to our knowledge about the alpha. These should be alpha subunits here. Right. So let's look at that figure 4.6. Oops. Okay. So, this is looking at cyclase activity in lymphoma cells, and so it's adenylyl cyclase, and they're adding these alpha S subunits. Okay, so we have I'm trying to so they have they treat with a ligand these lymphoma cells, and most of the cells die, and they basically they're selecting for the survivors a couple times here, right? And once they get the lymphocytes that they want that don't have the cyclase activity, right? So they're basically selecting for the cells that don't have cyclase activity. This hormone isoprenaline stimulates cyclase activity, but they're selecting for these ones that are negative for cyclase activity. And then what they did is took the membranes of these cells that don't have the cyclase activity, and they saw that they could restore activity by adding in the alpha S subunit. Right. So these cells don't have, let's see, reconstitution. So the cell line fails to generate cyclic AMP in response to the stimulation of the ligands that bind to receptors linked to GS. But by adding G protein alpha S subunits, they can restore the activity. And I'm trying to understand this. I'm sorry, I can't remember what my point was with this figure. Oh. My brain. Go back to the notes. Well, I think in general, just what this was showing is that the importance of the alpha subunit in activation of the adenylyl cyclase. So they found cells that were unable to, that's what it was, cells that were unable to, they had the cyclase, but they were unable to stimulate the cyclase activity. That's right, that's where I was getting tripped up. So they chose for cells that have the enzyme, but the enzyme was not activated. So then they showed that when you add the alpha S subunit back, they could activate that enzyme. I'm sorry. So that was just the experiment, how they showed the importance of having the alpha subunit of the G proteins. And so there are 16 genes for the G alpha subunits. And then if you take into consideration splice variants of G alpha S and G alpha O, you get about 20 gene products. And so in our cells, most of them express 9 or 10 of these gene products. And some of these G proteins, like the GT, the G olfactory, and the G gustatory, are only expressed in single class of sensory neurons. So the alpha T is in the visual system. The alpha OLF is obviously in the olfactory system, and then gustatory is taste. So they're in like taste cells, right? Some that are expressed only in cells with common origin, this is why we don't talk about them that much, is the GO, the GZ, and the G16. But the ones that are universally expressed are the ones that Dr. Johnson talked about quite a bit, right? The GS, the GI, and GQ/G11. So let's look at the types and functions of the G alpha subunits in this table from the text. So if we go back here again. All right. So you can see they can be like grouped together based on their generation of second messengers. That's basically how they can be grouped together. So we have the alpha S, the alpha olfactory, the alpha 11, and the alpha 12. Alpha I1 and alpha I2. It looked like 11 to me. So you have alpha S and alpha olfactory here, these two. These stimulate adenylate cyclase, so we're going to get an increase in cyclic AMP. Where, I don't like this pen, these inhibit adenylate cyclase, so you'll get a decrease in cyclic AMP. The alpha I3 also inhibits adenylate cyclase, and I'm not sure why they keep that one separate from the others. Alpha O is going to be specific for one type of adenylate cyclase, the type 1 adenylate cyclase. And also, which I thought was interesting, is that these G proteins can also interact indirectly with other receptors, right? So in the case of alpha O, it potentiates the EGFR receptor and therefore RAS signaling through MAP kinase. It can actually regulate other G proteins by sequestering this G activating protein RAP1 here, and can thereby activate, or this GTP ACE activating protein. It sequesters it, so therefore leads to activation of STAT, because it's not allowing the GTP ACE activity to occur for those G proteins. And it can also, this type of G protein can also, indirectly interact with other types of channels, such as the L-type channels in cardiac muscle. So the alpha T, we're going to talk about this when we talk about the visual system, but this actually stimulates the production of cyclic GMP phosphodiesterase. So we'll talk about that more when we talk about the visual system. And then we have some that, like this alpha Z, that closed potassium channels. So you can imagine that would have an effect on membrane conductance, right? G alpha Q, we often think about just stimulating phospholipase C, but it also stimulates this other protein known as BTK. That's bromodomain tyrosine kinase, I believe. Yeah. And bromodomains interact with DNA, and they can affect transcription and translation. We have alpha 12 that interacts with BTK as well, but it can also indirectly affect the activation and deactivation of other G proteins by affecting these gaps and these GEFs. And then alpha 13 also can indirectly affect the activation of other G proteins. So the reason why I'm showing you this is because when we learn about G proteins, and Matt can attest to this, we focus on G alpha S, G alpha I, and G alpha Q. And those are the only three I really talk to about PharmD students. But I think it's important as a graduate students to understand there's these other types, because you never know if you're going to come across these in your experiments, right? If you're going to see these other types of G proteins. Okay. So let's look at 4.7 too. Sorry about that. So then to kind of lump these together according to their amino acid identity, right? We get these trees, these phylogenetic trees here that show us how these are related. So we end up basically with four main classes of G proteins. We have the GS, GI, GQ11, and G12. And this is another reason why we focus on these three main classes. And then you can just see how they work here, where they're expressed. GS and certain isoforms of GS or GI and GS are ubiquitously expressed. GQ, same thing. G12 is also ubiquitously expressed, and you can see that it interacts with these GEFs, so it indirectly also affects other G proteins. And then we just put them once they're grouped into these four main groups, we look at the generation of the second messengers and the effectors they work through. And we know now G alpha S works through adenylate cyclase by positively activating it. GI works through adenylate cyclase by inhibiting it mostly, but then in some areas, some specialized cells, it also activates this phosphodiesterase for cyclic GMP. And like I said, we're going to talk about that when we talk about the visual system. And then when we think about GQ, we think about phospholipase C. And G12 is unique in that it doesn't really have the generation of second messengers. It really just affects these other proteins in order to regulate signaling through other G proteins. Okay. They're calling this BTK. I thought it was bromodomain brutines tyrosine kinase, non-receptor part. Oh, so that's not what I was thinking of. Brutons tyrosine kinase. Yeah, I thought it was bromodomains. Never mind. Receptor. And we don't talk about that too much, so sorry. It's not true. Okay. So then there are, so we think about the receptors in a way such that they bind mainly to one class of G proteins. They're either going to bind G alpha S, G alpha I, G alpha Q. But there are some receptors that bind to multiple G proteins. And so these top, right. So there are some receptors that bind to multiple G proteins, just period. But then there are toxins, these two toxins that can dissociate or activate the G proteins. They can either inactivate or activate. So cholera is a toxin that works by promoting GS activation. So it does this by inhibiting the ability of the GTPase activity. So we'll talk about this in a minute. So that's going to affect one of those dissociation constants. And pertussin, promotes the inactive state of GI. So it keeps the GDP remains bound, so it keeps it inactive. So if we're talking about these two toxins, if we look at cholera toxins, promotes the GS activation by inhibiting GTPase activity. Which constant is this going to affect? The catalytic constant or the dissociation constant? Catalytic, right? So if it's going to inhibit that constant's going to decrease or increase? Decrease,
So it will
right?
stay in active state.
Right, exactly. Good. And then if the pertussin toxin promotes the inactive state with GDP remaining bound, is that going to be the catalytic or the dissociation? Dissociation, right? So that constant is going to go down, right? Okay, good. And then I had one more handout related to this part 1 of this chapter. Just give me a minute, it'll come up. So we want to look at, this is alpha subunits that interact with the membrane of the, so I already said that the C-terminus region of the alpha subunit is the one that's going to be tether it to the membrane. This C-terminus region also dictates its specificity for the receptors. So that's an important domain. And then the N-terminal region is where you get interactions with the beta-gamma subunits. Okay. So for binding of adenylate cyclase, the effector, there's 3 distinct regions on the alpha subunit that are necessary for binding to the the adenylate cyclase and to release those beta gamma subunits to go off and do whatever they want to do. So we have the RAS-like domain, which is similar across all the alphas. These are all alphas, alpha subunits, subtypes. So they have homology in this RAS-like domain, and the RAS-like domain is simply the GTPase domain. And then in the helical domain is more variable across the alpha subunits and will help to determine some of the specificity. And then with beta gamma subunits, there are 5 beta subtypes. There's 1 to 4 and beta 5. 1 to 4 are grouped together. And there are 12 gamma subtypes. And, oh wait, let me find my notes. Sorry. No, that's when we get to America. Sorry. Right. So here's some more notes. Sorry. So the C-terminal region of the alpha, we already said, dictates the specificity for receptor. This is also where pertussis toxin works. It to inhibit that reaction. And then the N-terminal region with the beta-gamma subunit. Right, so there's a addition of a fatty acid to this region, to the N-terminal domain, the N-terminal glycine, and it helps that promotes the interaction with the beta-gamma subunit, so to promote that heterodimer formation. Oh, here it is. 3 distinct regions are necessary for binding to adenylate cyclase and release of the beta-gamma subunits, and they are the SW1, SW2, and SW3. They then undergo a conformational change with the exchange of GDP for GTP, and that's what facilitates the binding to the effector enzyme. So these SW1, SW2, and SW3 domains, they change conformation, and that is what is the signal for the effector enzymes to bind to the alpha subunit. Okay, yeah, and here's these domains. There's the RAS-like domain, and that's similar, like I said, across all alpha subunits. Contains the GTP binding site, that makes sense. And it has binding places for the beta-gamma subunit, receptors, effectors, and the regulators of G protein couple signaling. And then there's a helical domain that's more variable across the alpha subunits. it covers the GTP binding site, and this is where what undergoes conformation change to allow GTP dissociation, and this helical domain aids in GTP hydrolysis. And I think that figure is shown pretty nicely in the first one we looked at with this here. Here's this SW1, SW2 domains that obviously can link the beta gamma subunits to the alpha. Here's the helical domain, and here's the RAS-like domain up on the top. Oops, sorry. Go away. How do I get the? Why won't this go away? Oh, there it is. Right. Okay. All right, and then the beta gamma subunits. There's 5 beta subtypes. So the beta 1 through 4 are grouped together because they share a lot of homology and they're pretty small proteins. The beta 5 doesn't share as much homology with the other subunits, about 53% it says. And this is the one that associates with GQ. So beta 5 is the one that with alpha Q. And then there's 12 gamma subunits. They're small proteins and they're much more diverse. They don't share as much homology. And what's interesting, I thought, is that not all combinations of beta and gamma exist. So you might not find beta 1 with gamma 1. You might not find beta 1 with gamma 2. So not all the combinations exist. The gamma subunits are post-translationally modified such that they get hydrophobic groups added to them once they are translated, and that's what helps them tether to the membrane through that C-terminal domain. So this is also what I thought was really interesting, and this was an assignment that I had in mind for us to explore. So we often, when we talk about G protein coupled receptors, right, we think about the G alpha protein. We don't think as much of the beta gamma. Mm-hmm. But the beta gamma subunits are also signaling proteins. And the way this was found originally was in oocytes, which are frog eggs. And what they do is they express the beta gamma subunit and the potassium channels. And they saw that the beta gamma subunits could inhibit the potassium channels such that you got what they quote unquote called a bradycardia, like something you would see in cardiac cells. In what?
Decreased heart rate.
Yes, decreased heart rate. That's right. So by inhibiting these potassium channels. So the assignment then here is to find some papers that have drugs that target these beta gamma subunits, right? Because we think about the drugs targeting the G protein coupled receptor. We know these toxins that target the alpha subunit of the G proteins, but we don't talk as much about the beta gamma subunits being targets for any kind of therapeutic indication. So that's going to be our assignment for what dates do we meet? I have so many classes. Do we meet Monday or Tuesday? Tuesday. So for Tuesday, the assignment is going to be for each of you to find a paper that has some kind of therapeutic indication that targets these beta gamma subunits. And you, if you can communicate with each other so you don't all show the same paper. Some of you might find some of the same papers, but that's okay. But let's communicate so you don't necessarily find all the same paper. Okay, so to me, I don't think it's important to memorize all this stuff or anything. I'm not going to test you on what's the homology of these subunits. To me, what was important here is just to add to the information that you already learned about the basic information about the G protein coupling receptors and then talk more about the specific structure of these receptors. And I think why I think that's cool, and like when I was first preparing for this class a few years ago and digging more into this, because I'm not a G protein coupled receptor expert by any means, but teaching this class has forced me to read through this stuff, which I think is cool because even if it's not directly related to your research, it opens your mind to thinking about proteins in a different way, right? Because you just learn about G alpha and you learn about G alpha S and G alpha Q and G alpha I, but the beta gamma subunit kind of falls to the side. But that might be an important thing that comes up with some of your experiments if you are working on, receptor activation. So I think in this way it forces us to think about, things that can happen outside of what we typically learn. Okay, and we'll do this. Okay, yeah, we're going to get into the G protein receptor kinases too later. Yeah, okay. So that's where we're going to end. On Tuesday, we're going to talk about what you found with this, with targets for the beta gamma subunit. And then we'll continue with how the G proteins can be regulated by these GERCs and GAFs and etc.
Dr. Carpenter, I have a question.
Yes.
So, in the textbook, there were some mentions like a full agonist, partial agonist, and then there was inverse agonist as well. So, in case of inverse agonist, is that same as the antagonist, partial antagonist, or close to the full antagonist?
So, agonist we know activates receptor.
Yes.
Right? Partial agonist, you don't get the full efficacy.
Yes, yes.
Inverse agonist actually is not the same as an antagonist because it causes the opposite. It causes a decrease in the reaction.
Right.
Antagonists have no action in and of themselves. Right.
When I saw the curve, dose response curve, it goes down like a
yes, it goes down exactly below.
Yeah.
So let me just put that up here. If we were to draw that, right, we would have here's a response, whatever response we're looking at, right? And here's a concentration of, whatever drug, right?
Yep.
Okay, so we have the first one that this is 100% response. That's a full-on agonist, so we get this, right?
Dr. Kavanaugh.
Oh! Thank you. Sorry, it went off. Oh, you all can call me Jane, by the way. Graduate students are allowed to call me Jane. And Matt. You're considered a graduate student when you're in this class. I tell all it. Okay, so that's a full-on agonist, right?
Yes.
And then we have a partial agonist, which might start out the same, right?
Yep.
Start out the same as this, but then it'll come here, right?
Yep.
Because we won't get the full-on response. This is partial. So then if we have an antagonist, it's just this, right?
Yes.
Because you get no response.
Right.
And it does nothing in and of itself. But then the inverse agonist would be this. Right? It decreases.
Yeah.
Right?
Yes.
Okay. So was that your question?
Yes.
Okay. Because it actually causes like an opposite effect.
Right.
But and often we think when we don't think, but when students often think when they're first learning this, antagonists act like this, right? That's not the case. The antagonists have no effect in and of themselves.
Right.
Okay. Them on Tuesday, and if you could send it to me by like Monday at noon, so I can put it. This time I will remember to put it up on Canvas for you guys, so we can all see the paper that you're going to present. Okay?
Yep.
Same way, 5 to 10 minutes.
Yeah. Anyway, try to get it done in one class so we can move forward and look at these gaps and these RGSs, regulators of G protein coupling signaling, and the GTPase activating proteins. Because those are also very interesting. Those could be interesting targets as well. And then I know in your syllabus it says about exams. I got to talk to Dr. Johnson how he wants to do that. Last year I gave these homework assignments of presentations. Those were a big part of the grade. And then I gave 2 or 3 exams, but they were all take-home exam type things. So I just want to talk to Dr. Johnson if he still wants to do it that way, or if we just want to keep doing these assignments and that's your grade. I'm fine with that too. We'll see how it goes. Maybe we'll just have one like take-home exam at the end or something, just kind of summarize the major